a Department of Biology, and Earth and Planetary Science. And in homage to this, what I would like to convey to an audience of chemists are two things. One, as I'm sure many of you appreciate, microorganisms are really the greatest chemists on the planet. And we know this because of the rock record that tells us that for billions of years of Earth history, they've been catalyzing truly extraordinary reactions that have transformed our planet. Now, in my lecture today, I'm going to be taking you on a journey from iron oxides to infections, which is somewhat of an unlikely pathway to travel. And so I thought I would begin by telling you a little bit about a way we think about biological problems in my group, which is motivated in large part by an Earth historical perspective. So we heard earlier this morning about climatic change in the context of thousands and millions of years of Earth history. And surely understanding past records gives us insight into what is currently happening today to be able to give us a perspective on those changes. But, of course, processes that occurred not only in the past, shaping our thinking about the present, but processes that today occur, be they physical, chemical, biological processes, can give us insight into the past. And so this figure here is a picture taken from a hike in South Africa, where what you're looking at in the foreground are rocks that are 3 billion years old. And in the background, you see a sediment that contains ripple marks. It was a wave crashing on a little beach, dried up, and when we went on this hike, we snapped a photo because it was such a beautiful illustration of this principle of uniformitarianism that pervades thinking in the earth sciences, where we have a dialectic often intellectually between trying to understand how modern-day processes work with the assumption that these processes are very conserved through time and may give us an ability to interpret past phenomena. And then, of course, the reverse. And so what I'm going to specifically talk about today is how our work looking at microbial interactions with minerals led us down a pathway where I'm quite convinced some of the fundamental processes that we were thinking about in the context of microbe-mineral interactions may also be very informative in thinking about how we treat certain types of anaerobic infections. So to begin with a timeline of Earth history that now goes back many billions of years to the origin of the Earth, we know that microbial life has been around this planet for almost all of its history. In fact, we have isotopic evidence from carbon isotopes that tells us life was present at least 3.8 billion years ago. And we have a number of other ways of assessing the antiquity of life. And I've grouped them here into three primary categories. Morphological biosignatures that can come in various scales, from that of millimeters to microns to even nanometers. Here you see biogenic particles of magnetite forming inside a cell that makes magnetite to help it navigate within the geomagnetic field. We also have molecular biosignatures, both isotopes of various elements and more complex organic structures that incredibly are preserved in rocks as old as 2.7 billion years old, which we know must be derived ultimately from some form of life. And finally, we have a genomic record, the fossil record of DNA. And from this, we can begin to deduce evolutionary relationships not only between organisms, but also proteins that these organisms contain that catalyze very critical reactions that shape the planet. Now, for me, thinking more as an environmental scientist, I like to consider this a fossil. Now, you might not think initially that this is a fossil because it's a huge ore mine. And in fact, this particular ore mine comes from Western Australia. This is the Hammersley Formation. And it's also known as a banded iron formation because of the type of mineralogy present in this deposit. Now, we have a record of these types of formations that go very far back in time, extending back, at least to around 3.8 billion years, and continuing throughout Earth history. And what is now, I think, very well accepted is that the root of these formations in terms of the catalytic agent that led them to be precipitated is microbial. And here's how it works. So back in ancient times, there was quite a lot of ferrous iron dissolved in seawater. And something catalyzed its oxidation to the ferric state, which then rapidly speciated to form different types of iron oxide minerals that then went through the process of diagenesis to result in these deposits that today we can observe. Now, the catalyst, one of the most important catalysts for this process of iron oxidation, was some type of microorganism performing some type of photosynthesis. And there's a debate over whether the type of photosynthesis was oxygenic, meaning taking, as I'm sure we'll hear a lot more about this later in the afternoon session, water to molecular oxygen, and then that oxygen chemically reacting with the ferrous iron to oxidize it, or alternatively, anoxygenic photosynthesis, where ferrous iron itself was a substrate for oxidation powering photosynthesis. Now, be that as it may, either one of these microbial processes helped bring about the formation of these minerals. And then once they arose, this provided a fertile substrate for respiration, the opposite process, where these iron oxide minerals could serve as electron acceptors, much as we use molecular oxygen today, allowing the burning of organic carbon. And this is the part of the story that I'm going to focus on and explain how we got from trying to understand the details of this process of respiration to some insights that are relevant for infection. So we began our work by studying a bacterium called Shigella onidensis, and this is a scanning electron micrograph that you see here. This is a really wonderful organism because of its metabolic versatility. So it can respire oxygen, and in addition, many other substrates, including iron oxides and manganese oxides. Now, as you can see here, the cells are forming aggregates, and some of them are directly touching this ferric oxide below. And from a cell biological perspective, how these cells are acquiring energy from growing on this ferric oxide is a very fascinating problem. And here's why. These cells contain two membranes. And let's focus first on the inner membrane, which is also called the cytoplasmic membrane. This is where the cell is able to generate a gradient across this membrane that enables it to power the formation of ATP within the cell. This is called oxidative phosphorylation. And the way it works is that within the cell, there is some substrate that eventually gets oxidized, providing electrons to a membrane-bound enzymatic system that can couple the translocation of proteins across the membrane to the transfer of electrons through the membrane. And this occurs through various carriers, both proteinaceous and small molecules such as quinones, until those electrons are received by some terminal electron acceptor. Now, in our cells, in our mitochondria, this electron acceptor is oxygen, and that also can be the case for Shigella. And if it's oxygen, it's not so difficult to envision how this happens because we know that oxygen can diffuse across the membranes to the site within the cell where it is reduced. However, iron oxides are a totally different story because under the pH that's relevant to the problem here, they are very, very sparingly soluble, effectively insoluble. And so how does this electron transfer process work? They simply cannot diffuse within the cell. We now have a schematic for how this works based on a lot of genetic and biochemical experiments that have been done in a variety of labs, and I'm summarizing this for you here. What we now know is that the cell directs electrons to iron outside of the cell, again via a series of electron carriers, many of which are C-type cytochromes. And the part that I want to focus on is what happens at the interface between the outermost portion of the cell membrane, where there are some decaheme C-type cytochromes, and the iron oxide mineral itself. The actual molecular details of how electrons get from these proteins to iron 3 to reduce it to ferrous iron are still the subject of much active research. And one thing that has emerged is that this transfer is likely mediated by small molecules in the environment, such as humic substances. And I'll show you their structure in just a minute. But the general idea is that this is a process that we can call extracellular electron transfer mediated by exogenous electron shuttling molecules. So imagine you have shuttle molecule X. it gets reduced by the cytochrome on the outer membrane. Then in its reduced state, it can transfer electrons abiotically to the iron, reducing the iron and then itself becoming reoxidized, so it can serve as a catalyst. Now, humic substances were first proposed to be very important by Derek Lovely and his colleagues who were working at the USGS in Reston, Virginia. And what they found was that spiking groundwaters where microbial iron reduction was occurring with these macromolecular structures that constitute a very important fraction of the dissolved organic carbon in the environment, they were able to stimulate greatly the rates of iron reduction in those aquifer sediments. Now, the reason this is thought to be the case is because humic substances contain quinones, and these are the most important redox-active moieties on these molecules. So because of this, we can simplify our study of extracellular electron transfer in the lab by utilizing a surrogate compound called anthraquinone disulfonate, whose structure is shown here, that has the convenient property of turning color upon reduction by 2 electrons and the addition of 2 protons. Now, I'm a bacterial geneticist, and so as someone interested in the mechanisms of how bacteria catalyze reactions, this color change is very useful because it allows you to do a genetic screen where you can very easily isolate mutants that no longer have this capability. So again, taking this substrate as the indicator for extracellular electron shuttling, we first looked for mutants that had some insertion in a position of their chromosome at random and only one insertion in any given well, and isolate those that could no longer reduce a QDS to the hydroquinone, which stayed clear. And we found many different types of mutants in this process, but I'm going to only tell you about one for the sake of this story. And this one had a defect in the transport of a QDS. Now, how did we know this? Well, based on our analysis of where the transposon insertion in our genome lay, we noticed that it had fallen in the middle of a gene called TOL-C, which from previous research was shown to encode a protein in the outer membrane of many bacteria that was essential for the secretion of antibiotics and various detergents. So when we had this disruption in the middle of this gene, it essentially knocked out the capacity to produce this protein. Anthraquinone disulfonate built up in the cell, and this caused toxicity and therefore led to this apparent phenotype of a defect in reduction. So we were surprised by this because initially we hadn't made the connection between antibiotics and these electron shuttling compounds. But the more we thought about this, the more obvious the connection became. And you can see this when you just draw a few representative structures of known electron shuttles, such as AQDS, and compare them to important antibiotics, such as tetracycline, doxorubicin, which is used in anti-cancer therapy, and piocyanin, a natural product that I'll come back to in great detail in just a moment. The reason we knew that they were functioning the same way was because when we compared the response of the cells that were grown on a lawn on a plate, here that's what you see in the background here, and looked at the wild type version of these cells and compared their behavior to the mutant that couldn't make this important efflux channel, the TOL-C mutant, what we observed was that regardless of which compound we spotted on these discs that we put down on the surface of our plates, in all cases, the mutant responds similarly in that it was unable to grow within a certain zone of clearing of the molecule on this disc. So in this case, we were seeing that these electron shuttling compounds, such as AQDS, were effectively acting like antibiotics. Now, what this suggested to us, however, was that the opposite also would be true, that possibly redox-active so-called antibiotics that the cells were making might also be able to function as endogenous shuttles. And so the idea here is that the cell might be producing some molecule, excreting it from the cell, which could take the place of these other exogenous shuttles that I spoke about earlier. And the more we thought about this, the more it made sense, because if you take a look back again at how these organisms actually grow in the environment, they're growing as biofilms, and that is communities of cells that are attached to a surface. And as these biofilms develop, as you can see, not all of them are in physical contact with the surface below. Here you see some on top, and there are many embedded in the middle. So now just to draw a cartoon version of this and flip it on its side, this really illustrates what I think of as the respiratory problem that biofilm cells face, regardless of what surface they're attached to. And that is that cells in the middle are quite literally stuck between a rock and the hard place in terms of expending their cellular reducing power. They don't have access to oxygen because that's being scavenged more rapidly by the cells at the top than it can diffuse. And they don't have access to this terminal electron acceptor below, the iron oxide, because they're not in direct contact with it. So we postulated that precisely in this context would be where the ability to produce your own molecule that you could cycle might be very beneficial for survival. So to test this idea, we came up with a very simple assay, and it works as follows. What we designed was a 3-layer plate. Here you're looking at it from a bird's eye view. Here I'm going to walk you through it from the side, where what we supplied was in the middle a layer that we created of nutrient agar impregnated with manganese oxides, and above it, a very thick layer through which no bacteria could swim. And the beauty of manganese oxide is that under these conditions, when it gets reduced, it turns clear. And so this would allow us to very easily detect the production of some type of molecule, rather the activity of some type of molecule that was diffusing through the agar, enabling manganese to be reduced. So if you look here in green, what you see is this activity happening for Shionella onidensis. But on the left, there's even more activity for this organism, which is a strain of Pseudomonas aeruginosa that's very important in the context of cystic fibrosis. So this really caught our attention, and it's here that we began to think about this in a more clinically relevant context. So as I said, Pseudomonas aeruginosa is a very important pathogen for individuals with the disease cystic fibrosis. And the reason is because over time, within the lung of CF patients, mucus develops, and within that mucus, it's a very fertile home for the growth of bacteria. And as the disease progresses, Pseudomonas becomes the most important pathogen that colonizes the lung and ultimately leads to the death of these patients. Now, for over a century, microbiologists have known about Pseudomonas' ability to secrete beautiful pigmented compounds of the class called Phenazines. So this is their basic structure, and they come in literally all the colors of the rainbow. It's a very aesthetic organism to study because of the beautiful pigmented small molecules that it makes. And depending upon which particular chemical substituents we have at various positions around the ring, that will tune the properties of the molecules up and down the redox potential scale. It will affect their hydrophobicity and philicity, and of course also their color. Now these compounds are versatile redox-active compounds, and we know that also from just very simple visual cues. What you see here is a culture where cells have been grown up overnight. What we've done is just vortex it for 5 minutes on the bench. You can see the bubbles at the top, and it turns this lovely aquamarine shade. If you then let this tube sit on the bench for another 5 minutes, everything below the water-air interface turns back to a more yellow color, which is the background color of the medium, and it's only at the very top that we still have this blue color. And the reason for this is that these phenazines, collectively, when they're oxidized, exhibit a blue color, and when they get reduced, they go clear. Okay, so I said that phenazines have been known to scientists for over a century, and much of the attention that has been given to these molecules has focused on their antibiotic activity. And not surprisingly, it's very warranted, and that's because when reduced phenazines react with molecular oxygen, they generate superoxide radical species that are very broadly reactive and can do a lot of indiscriminate damage to cells. However, there's something very important to keep in mind when thinking about these compounds in terms of the biological context in which they're produced. And that is that they are made by cells only at very late stages of development, oftentimes when no molecular oxygen is present. And the corollary to this is that under anaerobic conditions, toxicity is greatly diminished. And so thinking now again more like an earth scientist, something that we also know, of course, is that oxygen has not been around the planet for all of its history. And we know furthermore that derivatives of these compounds can be found in the membranes of methanogenic archaea, which are a very ancient life form that certainly predated the rise of molecular oxygen on the planet. So even from a biosynthetic perspective, we know that the ability to make these phenazines is not dependent on oxygen. And because of this, we wanted to reevaluate whether their function was really an antibiotic one for the cells that produce them under the conditions that are relevant for their production. Now, Another conventional wisdom is that they are secondary. And, of course, by putting it in quotes, this clearly tips my hat, which is I feel this is a pejorative term and greatly unfair, because they're only called secondary because of the way bacteriologists have classically grown microbes in the lab, and that is in batch culture where we study growth, here looking at cell numbers rising over time in hours. And this phase of growth is what we call logarithmic growth phase or exponential growth phase. And when they start to plateau, when bacteria hit their middle age to old age, this is what we call stationary phase. And there's been a lot of attention by microbial physiologists on the adolescent phase of bacterial growth, but very little attention paid to their old age. And we can discuss why at the end if people are interested in that. But it's here, in this so-called stationary phase, that we suddenly see the burst in production of phenazines. And I've shown for you now two, pyocyanin, a blue phenazine, and PCA, a yellow phenazine. So we had a different perspective, and that was that under these conditions where oxygen is depleted from the cultures, phenazines might actually be key to survival. And so a way we decided to test this was to do an experiment in an environment devoid of oxygen, and provide an electrode to reoxidize the phenazines. So we constructed a mutant strain that couldn't produce any phenazine on its own, and we were able to add a discrete amount back to the culture that we knew was a very relevant amount, because this is roughly what the organisms will produce in the context of the lung. So here's what we did. We have this electrode set up. We have cultures growing. We have an electrode that can reoxidize them. And what Yuwen Wang, a postdoc in my lab, who was a student in Alan Stone's group, he's also here at this symposium as a Dreyfus recipient, did was look to see how the viability of these cells was affected by their ability to recycle phenazine. So within a period of a week, if they have pyocyanin and the electrode is turned on, they remain viable. They can form a new colony when they're plated. However, if you either turn off the electrode in the presence of the molecule or you do not add the molecule at all, you get a very dramatic drop in viability over this very short period of time. So this we thought was quite interesting and relevant, again, coming back to this cartoon, because the context where this matters for the organism Pseudomonas aeruginosa is in the lung, where they form biofilms, and the mucus that they inhabit has been shown to be hypoxic through microelectrode measurements. Now, how can we test whether or not this idea bears out in terms of the development of biofilms? Everything I showed you so far was looking at growth in planktonic liquid culture. Well, we can begin by doing some very simple experiments by growing biofilms on the surface of agar plates and monitoring their development over time. So here I'm showing you a comparison between a mutant that can't make any phenazine, the wild type, and a strain that overproduces phenazine. And we're going to now go through from day 1 to 3 to 7. And what is very obvious is that whereas the mutant that overproduces phenazines remains nice and smooth, the mutant that can't make any at all flattens out, expands its surface area, and becomes very wrinkled. So what's happening here? It's actually very, very complex, and I'm only going to talk about a small portion. So there are at least 3 different ways phenazines can be influencing this phenotype we see. The first is that they're signaling molecules that direct a specific behavioral response by binding onto transcription factors that recognize them, and we know that this occurs. The second is, of course, that the phenazines are affecting cell surface properties by binding to the cell, and that affects their ability to interact with each other and influence the morphology that we see. But the part that I want to talk about in the remainder of my time is their ability to function as oxygen surrogates, which itself might elicit some type of signal within the cell that shapes the behavioral response that we observe. So the specific question I want to address is whether the wrinkling phenomenon we saw with the mutant that couldn't make any of these electron shuttling phenazine molecules, is that consistent with an oxidant access strategy, as I've been building up to in the talk? So we might imagine that for the overproducer, it would be able to make a nice smooth biofilm because in the zone where oxygen occurs, it can use oxygen as an electron acceptor. That's shaded here in blue. But in the anaerobic portion of the biofilm here in white, it would be able to get by because it's shuttling these phenazine molecules. However, the phenazine mutant, you might predict, would spatially organize itself so as to access oxygen everywhere because it wouldn't have the option of recycling its own endogenous shuttle. So towards this end, we used microelectrodes to measure where in various strains the threshold between oxygen availability and anaerobiosis set in. And we found everywhere we looked that at a depth of approximately 200 microns, oxygen went to 0. So let's now take a look at these colonies in a little bit higher resolution. So if you're at a breathing room in Los Angeles, the idea is to spread out. Of course, you know the joke coming next. In New York, it's to build up. And if we now measure quantitatively the dimensions of these beautiful films, this is of the mutant, recall, that can't produce any phenazine, what you see here that's so lovely is that even at the maximum thickness between any 2 points, it's no greater than approximately 400 microns, which is consistent with the idea of oxygen diffusing in from any given side to a depth of about 200 in either case. So the prediction from this would be that if we were comparing the wild type to the mutant in terms of the depth to which cells could grow, we would see a difference in depth. And that's what we observe. So indeed, when we look here using transmission electron microscopy and measure where the viable cells are residing, we see in the wild type a much greater depth than we see in the mutant, which, recall, is unable to excrete these phenazines, whereas the wild type does. So this feeds the idea. And more importantly, we're also able to observe that it's really the redox balance that is being affected in this morphological response. Because if we compare now the morphology of the wild type to the mutant that can't produce these molecules to a different mutant strain that is defective in the ability to reduce the phenazines, it still can produce them, but it can't reduce them, we see a very similar phenotypic response. It's essentially choked. Even though it has the molecule there, it can't use it. But if we rescue it with the addition of a different electron acceptor so it can restore its redox poise, such as nitrate, it restores the wild type phenotype. And the same thing is true for the mutant if we add either nitrate or piocyanin. So just to end, what I'd like to leave you with is the idea that this phenomenology that I've talked about in terms of Pseudomonas aeruginosa may be very widespread in nature. And some of the evidence we have of this comes from work we've done with Streptomyces silicolor, which is one of the most important antibiotic-producing organisms in the soil. Here you have a picture of actinorhodin. It's this beautiful blue pigment being produced by the streak of bacteria on the plate. And what we, again, were able to do was compare the development of biofilms of mutants of Streptomyces that were unable to make this redox-active secondary metabolite to the wild type. And again, we have a very similar phenotype. It's slightly different because this organism can produce spores, and that's the white stuff you see in the middle. But in terms of how it flattens out and wrinkles up and maximizes its surface area, it's identical to what we observe in a very evolutionarily distant organism, Pseudomonas aeruginosa. So just to conclude, the take-home message, I hope that you find of relevance to the environment in many different environments, is that when microorganisms are growing as multicellular communities on any surface, be it an iron oxide, a lung surface, a plant root, or an electrode in a marine sediment, in all of these contexts, as they develop beyond a certain critical size, they will become limited for oxygen. And it's in these type of environments that the ability to produce these so-called secondary metabolites might actually be absolutely essential for sustaining their metabolism. So with that, I'll end and acknowledge the people in my lab who did much of the work that I spoke about today. Here's Yuan and another postdoc in the lab at Cell Ramos Solis. They've both been working a great deal on the biofilm aspect of this. My student Mario Hernandez began the work with iron respiration Shewanella, but the two that have made the most important contributions have been a student from Caltech, Alexa Price-Whelan, and Lars Dietrich, a postdoc in my lab. And I've been very lucky to work at two great institutions, Caltech and more recently MIT, and also be supported by the Howard Hughes Medical Institute. So thanks very much, and I'd be delighted to answer questions.
Questions? Re-identify yourself. Don Stedman again, the University of Denver. Your picture of this layer on the lungs showed the lung surface to be completely devoid of oxygen. That strikes me as a bad idea. Do you have thoughts about how that might be alleviated?
Well, so this I should say is the lung surface of cystic fibrosis patients. Okay, so this is not the typical lung surface that I have drawn. And the problem with the CF lung epithelial surface is that in the disease, there is a mutation in what's called the cystic fibrosis transmembrane conductance regulator. And that's a cyclic AMP-dependent regulator of sodium channels. And in the absence of a wild type, a normal version of this, this protein, sodium absorption accelerates, and because of this, sodium and potassium ATPase pumps aren't inhibited anymore, and so it leads to a large increase in oxygen consumption by CF epithelial cells. And so that's something different going on in the context of the lung than in a normal lung. Another important difference that's been shown recently, getting back to iron, is that for some reason that's not well understood, these mutated CFTR lung epithelial tissue cells excrete a lot more iron. And independently of anything I've told you, iron is a molecular signal for biofilm development. And so then it gets compounded because the cells colonize these epithelial surfaces better, they form biofilms, they scavenge more oxygen, and so all of this contributes to a very hypoxic environment.
Chris McNeil, Chris McNeil, University of Minnesota. I enjoyed your talk, Dianne. I was curious about these quinones. You mentioned that quinones are naturally occurring in the natural organic matter milieu, and that these bacteria can also produce quinone-like molecules. Is there any sense for whether one can come from the other? In other words, the stuff we find in humic substances, is there any sense that those are coming from these soil bacteria, or
yes,
are the...
They are, and that's been shown by many others in this field. One of the best examples is the work of Dianne McKnight at the University of Colorado at Boulder, I believe, right, Francois? So she's been saying this for years, and it's been shown that depending upon which environment you isolate the humics from, they're either derived from the degradation of plant matter or microbial matter. Natural products that are produced by plants and microbes together wind up coming together in the soil to form these really complex structures. So
so I guess the question I had is a little bit more specific, which is that the so-called terrestrial organic matter, which is largely derived from soil, the vascular plants contribute to that pool, of course, but then you also have microorganisms in that soil as well. And to what degree are the microorganisms responsible for the terrestrial component?
Oh, I see. I don't know quantitatively, no number to put on it, but I would, of course, and I'm sure you would agree, assume that the microbes, through an indirect way, are very much affecting the ability of the plants to excrete what they're putting into that terrestrial environment by providing them with critical nutrients, for example, even if it's not through some direct transfer of these molecules from one cell to the next.
Charles Dismukes, Princeton. Very nice talk. And I don't know much about these particular microorganisms, but I know others, the phototropes, the microbial ones, that if you add molecules like this, you actually catalyze the production of superoxide, and that kills the cells. Whilst here, you're talking about endogenous antibiotics that are actually rescuing the system. So I presume that death by addition of exogenous antibiotic could occur by an oxygen activation mechanism as well, although I don't know that's the case here. But is it the case, and how do you separate that death from the
It very much
rescue?
is the case. And what's really quite interesting is that these organisms have evolved very complicated strategies to protect themselves against their own... Oh, sorry. Okay. Thanks, Francois. So the very interesting thing is that these organisms have evolved many complicated strategies to protect themselves against the generation of superoxide by their own endogenous molecules. And it includes producing proteins that specifically sequester these molecules within the cell to minimize their activity. The other thing is there are specific efflux systems that recognize these molecules that tightly control the amount that's within the cell. And the third is that they produce extra copies of superoxide dismutase to help quench any reactive species that escape those other two mechanisms. Now, if you add a different compound, it's not the endogenous one, like methylviologen, for example, then they die. Then they're in real trouble because the enzymatic machinery has evolved over the years to be very specific for the ones that they themselves make.
One or two more questions before lunch, perhaps. That's some questions. We just need to Dianne afterwards. Thank you, Dianne.
Thank you.